Nikita, only 30 seconds. Wait, only
okay,
30
sir.
seconds.
Sure, sir. Sure.
Yes. So I have started YouTube. Now I will share. And
yes, sir. Webab, sir, you can just check that if you can share the presentation.
Yeah, sure. Do you see my screen?
Yeah,
now?
it's visible, sir.
Okay. Do you see full screen or everything is fine, right?
Yeah, you need to go for the slide share.
okay. It's a full screen. No problem.
Yeah, okay. Because I tried yesterday and it wasn't somehow working when you
No
go to
issues, sir.
screen. So I think.
No problem, Nikita. You can start.
Okay, sir.
And I request all others. Dr. Nirmala has joined. Okay. Dr. Ajay, please introduce yourself. I'm very happy to see you today. Please introduce. Dr. Ajay.
Okay, sir. Good morning, sir. Good day to you. Yeah, good afternoon.
Yes.
I'm Professor Ajay from Adekunle Ajasin University in Nigeria. I'm currently the Director, Center for Infectious Disease Control and Development. And it's a pleasure to come online with you for this particular presentation.
Thank you. Thank you, Nikita. Proceed.
Yes, sir.
Thank you, sir. So good afternoon, everyone. Good morning and good evening to everyone. So today we have a session at 5:30. It started at 5:30 today. So there's a different timing because we have our speaker from Germany today. So hi, everyone. I'm Dr. Nitika Thakur, and I'm working as Assistant Professor in the Department of Biotechnology, Schulne University. Today, I'm very happy and glad to welcome our speaker from Germany, Mr. Webab Dolare. And I extend a warm welcome on behalf of MBSI for the speaker today. And he'll be talking about a very important topic today, to the technological transformation of pharma microbial sectors. So before we go further with the lecture, I want our President, Professor Deshmukh, to just share the mission and vision of MBSI. So over to you, sir, for sharing the mission and vision.
Thank you, Nikita ma'am. Really, I'm very happy. Just Nikita ma'am introduced me about Vaibhav sir, and I'm very happy that he is my younger brother. In the sense, we studied in the same institute, and he is an expert. And although he is an expert, he is working well in European countries. Even then, he is eager to share his knowledge with the original Indians. So it is a best opportunity in biotechnology to learn something from Vaibhav sir and to educate our student. This lecture is recorded in the form of YouTube, will be circulated all. I really appreciate efforts of Nikita because she is continuously taking efforts and arranging such lectures. Thank you. Thank you.
Thank you so much, sir, for sharing the mission and vision of MBSI. And I welcome Mr. Webab Dolare today to this session, to this lecture session, Indian speaking from abroad. So Mr. Webab Dolare is presently working as Global Head of Quality, Novartis, Germany. He's responsible for the anti-infectives and sterile manufacturing sites based in Europe. He has necessarily managed various transformation projects in Novartis, and he started his early career in biotech research and development in India in 1994, worked with India pharma companies in microbiological QC labs like Lupin Limited and Dr. Reddy's with increasing roles. He holds an enriched experience of QMS, regulatory affairs, and health authority inspection management. He has proven expertise in sterile aseptic manufacturing and quality. He has completed his master's in microbiology from Government Institute of Sciences, Aurangabad, Maharashtra. Today, he will be enriching us with technological evolution in pharma microbial focus. So over to you, Webab, sir.
Thanks, Nikita, and thanks Microbiologist Society of India. It's It's a moment for me where I'm really glad to be associated with Microbiologist Society of India for two reasons. One is the word India in that, and the second is the microbiologist. Both the words are really very important for me, being an Indian, being a microbiologist. And now I learned that Deshmukh sir is also a student of the Institute of Science. I'm really happy to be part of this lecture series. Thanks a lot. I thought maybe it will be good to talk a little bit about the technological evaluation in the pharma industry. As you know, microbiology is a core subject, important subject for the pharma industry as a whole. And it's important for the students. And I understand from Nikita that there will be quite many students participating in this lecture series. So I thought, why not choose this topic, which is more relevant for students to understand what they're learning today and how they're going to apply this knowledge when they enter into pharma industry, for example. So let's get started. I've divided my lecture into a couple of parts. So first, I will give you a brief overview about how the pharma industry look like globally and locally. When I say locally, in India, because it's important. Maybe there are also some people who are already working in pharma industry who are participating in today's lecture series. For them, probably this is repetitive information, but for those students who are still studying and they want to make the career, probably this information will be very useful for them. Also, then comes the role of a microbiologist, what typically a microbiologist does in a pharma industry. I'm going to be focused on the pharma industry only because there are various aspects differently done in a biotechnology industry, in the research field, but I will really be focused on pharma industry as such. Then I will explain a little bit about how it is evolving, the conceptual part of it and the technical part of it. When I'll go into the technical part, I will give a few examples about how the technology is evolving. Then I will also speak about the testing paradigm shift, how the testing has been changing over the years. And of course, at the end, as Nikita madam has suggested, maybe I will speak a little bit about the career guidance before we end up with the summary of today's lecture. So hopefully it should take about 45 minutes if I'm fast enough, and then we will have some time for question and answer if there are any. Okay. So let's get started directly from the scope of the pharma industry globally. So it's about 1.27 trillion US dollar. So I'm giving these numbers, don't get overwhelmed with these numbers. It's only to understand the scope of the pharma industry globally, how huge spend they're making on the research and development, how this industry is technologically driven. At the same time, it is highly regulated industry. I think this is very important thing to understand if we are thinking about working in this industry. It's highly regulated. There are quite a stringent regulations stipulated by different developed nations as well as in India. These regulations have to be followed when we are manufacturing the product. So this is very, very important point to be noted. Also important is to look at the employment. So there are more than 5 million people employed globally. So these figures are from 2020. I've particularly chosen this because these are the numbers from let's say during the Corona time where the overall industry was affected. But this is overall the scope of pharma industry, which is quite big. This is important to understand. Technology driven, a lot of spend on R&D. When I look at the Indian pharma industry, of course, Indian pharma industry is a part of the global pharma industry. The revenue is close to 38 billion. It was in 2020. What is important and peculiar about Indian pharma industry that we are the largest provider of generics drugs. I hope everyone knows generics. Those products or those drug products which are off-patented are called as generics drugs, and we are the largest manufacturer of generics drugs globally. Indian pharma sector supplies over 50% of the global demand of vaccines, and I think this is very, very important point. We should make a note of it. During the COVID-19 corona pandemic, Indian vaccine manufacturers were in high demand. So not only those who were doing research and trying to bring in the new vaccine, but also other who were purely involved in the manufacturing of vaccine. I think from both point of view, you have contributed very highly contributed in the form of vaccine manufacturing during the corona pandemic. I think this is very important and we should be proud of it. 40% of generics demand in the US and almost 25% of all medicines in UK are manufactured in India. Globally, India is ranked 3rd by production and 14th by value. If you look at the domestic pharmaceutical industry, then it includes close to 3,000 drug manufacturing companies, and they are having close to 10,000 manufacturing units. Of course, they are small, very small, like, employing 50 people, and quite a big, like employing 2,500 people and so on. So the range is quite wide, and that's why you see also the number quite big here. Okay, so that's about pharma industry in India. Now, let's get into the role of a microbiologist. So what a microbiologist does. I understand there are a few people already working in pharma industry who have joined this lecture series, but quite many of them are students, they're studying, and for them it's important to understand what is the role of a microbiologist. Because we learn so many things in college while doing our bachelor's degree, our master's degree, but when we step into industry, the world is different. So, that's the reason why I thought maybe it's good to give an idea of what we do as a microbiologist in the pharma industry. Basically, the structure of the pharma industry looks like this. We have receipt of raw materials up to the dispatch of the finished goods. So from the start of the process to the end of the process, there is a role for a microbiologist to play. So we typically have a warehouse, we have production planning, engineering, maintenance, supply chain. Most important is quality control, quality assurance, which has overall responsibility to look after everything that's happening within the GMP perspective. And also, of course, there are support functions like human resources, administration, finance, and R&D. Microbiology can be a part of the QC setup, or it can be a separate department depending on the product range. So for example, if we are manufacturing oral solid dosage form, that means the tablet, capsule, then typically the setup of a microbiology function is comparatively smaller. If we are manufacturing injectable products, then this setup grows exponentially. So that means we have a huge lab, we have too many people working in the microbiology, because there are a lot of activities to be done as a microbiologist if we are manufacturing sterile drug products. So the scope of a role of a microbiologist depends on what type of products the manufacturer is manufacturing in their premises. So that's about the role of a microbiologist. Now, if you go into more specifics, so what are the tests which are typically performed in a pharma company? So, of course, here you see the list, which is comprising of different type of product testing. So also the oral solid dosage form as well as the injectable products. The first one is the microbial limit test. Basically, it's about enumeration and identification and counting of microorganisms. Another important aspect is the water analysis, because we need water which is free of objectionable microorganisms. Also, the microorganisms which are controlled to a certain number, because we are producing products which are meant for patients, and they are supposed to be or expected to be free from microbial contamination. Then comes the microbiological assays, antibiotics, and vitamins. Of course, nowadays, quite many testing methods have been moved from microbiological assay to the HPLC assay, even for antibiotics and vitamins. But at least in the old days, when I was working as a bench microbiologist, I did a lot of antibiotic assay by means of microbiological methods, also for the vitamin B12 and so on. Then comes the sterility test. It's about testing an injectable product, because it is expected that injectable product is sterile, and this test ensures that the product which you're testing is sterile, that is free from microorganisms. Then comes bacterial endotoxin test. There are other tests like preservative efficacy test, typically used for the liquid products. Then there is also disinfectant efficacy test, because we need certain environment in the manufacturing facility, and that environment needs to be maintained free of microorganisms. And one of the way to keep that free of microorganisms is to use appropriate disinfectant. And those disinfectants have to have certain efficacy, and those efficacies are also tested in the microbiology lab. Another important aspect is that of environmental monitoring. It's also very important aspect which microbiologists perform, because again, here too, we have to ensure that the manufacturing environment is free of microorganism, or it is, controlling the microbial presence in a given area to a limited to a certain limit, which are defined by the regulation. So what are the typical activities we perform? So these are the tests, which are the, let's say, pharmacopoeial test, or the tests which are required for various activities and product testing and so on. But what are the activities behind it? So activities are like these tests, of course, but also media preparation. Microbiological media has to be prepared. This is also changing. Again, I will speak about this in the subsequent slide. But when I started my career, we also used to prepare the media on our own. I think we in the college and university still prepare, by adding different ingredients and then making those media. Of course, this is also changing. Nowadays, you get ready to use media, ready to prepare media, and so on. But this is also an important activity a microbiologist has to perform. Of course, the microbial culture maintenance, this is also something we still do in the universities. Of course, the environmental and personal monitoring, I just spoke about. We have to keep the people and environment free from microorganism. Of course, the laboratory maintenance in the universities, probably we have laboratory assistants who do that, but here as microbiologists, we have to do that. Then there are a lot of validation and calibration activities we have to perform. I think the most important part is also investigation and the expert opinion. I think here microbiologists play very crucial role, because the whole knowledge of microbiology, of if something goes wrong, if the product fails, if the water testing fails, if a microbial limit test fails, that's the result of the test. But what is behind that is the investigation. And in that investigation, microbiologist can only play crucial role, because he has the knowledge of the microorganism, the environment in which those microorganisms can grow, multiply, how we can get rid of those microorganisms, what are the sources of microorganisms, what are the root of contamination of those microorganisms, and so on. So here we play a very important role. And even though now I'm not working as a bench microbiologist for last 20, 15 years, but still, when it comes to investigation and expert opinion, this knowledge, this experience is helping me a lot. So I think this is very important, that our theory will not go away. Our theory will always be with us, and this will be helpful for us when it comes to investigation. Another part is training. We learn a lot of theory in university, but when it's come to actual job work or actual job or the bench work, the training, on-the-job training is very important. So this is also important aspect a microbiologist has to play. They have to also train others. So they have to also train the operators. They have to also train the supervisors who are working on the shop floor, to give them background about the microbiology. So what happens, because people who are working on the shop floor as an operator, they do not necessarily have microbiology background. So they do not know, if I do certain thing in a certain way, what is the impact of that action on the drug product. And that's where the role of microbiologist comes into picture, because they have to tell, if you do it in this way, if you're going over the product, you may shed some particles, and those particles may contain some microorganisms, and you may contaminate the product. So that knowledge is something which is expected from a microbiologist in a pharma company. And of course, not to forget, a lot of documentation. That's very, very clear that we in a pharma industry, as I said, we are a highly regulated industry, and that's the reason why we need to document everything that we do on day-to-day basis. So that's more or less about the role of a microbiologist in a pharma company. Now, let me switch the gear and talk about how the things are evolving over the years. And first, I will talk more about the conceptual part. If you think about the modern era of pharma industry, it dates back to 19th century, since when we have making the medicine, and since then the making medicine is ever evolving. We started, so if you think about India, for example, we talk about Ayurveda. So what Ayurveda is all about? It's about plant-based extract. So it was also the case for the modern pharmaceutical industry, where the initially plant-based extracts were being used. Then we slowly moved to the chemical synthesis, then fermentation, then came the vaccines. Now we are talking about monoclonal antibodies, gene therapies, and so on. So this is the conceptual evaluation of the pharma industry. So in prehistoric days, it was raw extract, powders, and liquids, which were commonly used as medicines. In the modern days, it's about making tablet, capsules, suspensions, syrups, injections, and so on. There were small molecules, antibiotics, and so on. If we talk about today and tomorrow, the future is more about monoclonal antibodies, recombinant DNA technologies, the CAR-T therapies, and radioligand therapies, and so on. So if you look at the new drugs for the difficult to treat conditions, it's more about the MABs, the CAR-T, and radioligand therapy. So this is how the evolution conceptually happening in the pharma industry. Then, if I speak about technology, so when it comes to technology, from yesterday to today, there were like no traditional way of manufacturing and testing. So you manufacture, you test, and you release. No, so that was the way of pharma industry operational model. There was a slow pace of technological development. Of course, there were developments happening. It's not that it was not happening, but the pace was slow. We were more relying on the people factors. More people, more production, that kind of mentality was existing. There was less investment. I mean, the idea was to invest less and get more output. That was the thinking process behind. And of course, not much focus on R&D. There was, of course, focus, but not much as it is today. When I think about today and what's going to happen tomorrow, we are more and more talking about continuous improvement approach, use of new technologies and automation. We make use of a lot of IT and AI, so artificial intelligence and information technologies being used on a very, very high scale in the pharma industry. We are talking about quite a many disruptive technologies. There is a lot of focus on innovation and, of course, the speed. Speed is extremely important, and we have seen this particularly when the whole world was facing this COVID-19 pandemic situation, where we wanted a vaccine tomorrow, and that was the speed required in the pharma industry at that point in time. So that's how the technological evolution is taking place in the pharma industry. Now, let me move into a more specific example. So we talked about how the concept has changed. We talked about how the technology is changing, and this I can explain you with some of the examples. So, and the first one, of course, is about, again about the COVID-19 or the corona pandemic which we faced a year ago. So we have seen there was a huge pressure on the pharma manufacturing, or particularly the vaccine manufacturers, during the recent COVID-19 pandemic. There was a need to produce more, because the whole world was affected because of the corona crisis. Everyone wanted to have vaccine. No one in the pharma industry assumed that we would need vaccine manufacturing capacities to that level, which was required at that point in time and in a very shortest period of time. This was a big challenge. Even the big companies were not able to, increase their capacities, because you need at least 3 to 4 years to build a new manufacturing facility, to put the things in place, to validate them, first of all, also to manufacture various equipment, instrument, to make the facility ready, to train the people, and to start the manufacturing. So it's a long process. And it was not possible to do that everything in let's say in few months, because we were talking about few months, even in few days. So we were more relying on how can we produce more from the existing facilities. And there was a need to produce more doses than the installed capacity. So for example, I can make only 100,000 vaccine doses a day, for example. But the requirement was probably 500,000, even 1 million or so. That was the scale of increase that was seen during the pandemic. Every time, and in the typical setup, you have to every time clean and sanitize the stainless steel bioreactors and to make the new batch. So this is the standard setup companies typically had, that you have a fixed kind of equipment which are made of stainless steel. You have certain defined number of campaign lanes. You produce one after another batches. Then after that, after a certain number of batches, you have to stop, you have to clean, you have to sterilize again, and then you have to start a new batch. That's the routine process. That of course takes time, because for cleaning, sanitization, you have to stop the manufacturing. When you're cleaning and sanitizing and sterilizing, you cannot produce. That's a downtime, we call it in the pharma industry. And this was not affordable in the pandemic times. So what we did, we use innovative technologies. We use bioreactors which are disposable, sterile, ready to use, disposable, large size bioreactors. So instead of conventional stainless steel, where the downtime is zero, you just bring in the sterile bioreactor, plug it, make the aseptic connection, you start manufacturing. So that is the one important aspect most of the manufacturers, the vaccine manufacturers, use to meet the highly increased demand of the vaccines during the pandemic. So this is one example of the technological evolution, where we move from fixed stainless steel bioreactors to sterile, ready to use, disposable, large scale bioreactors. So this is one example of the technology. When it comes to other technologies, for example, isolators and wraps, this is something evolving over the years. Until recent times, we were using zone A areas which are having specific filtered air coming into these areas, and we have laminar airflow benches. Probably most of your universities also have these laminar airflows, where you do a lot of manipulations of your cultures and so on. So these were the typical laminar airflow benches or the cabinets which were being used for the sterile injectable product manufacturing. And this was very common. This is even today quite common, I would say. But use of laminar airflow and surrounding clean room requires a lot of people interface. You cannot get rid of people. It's always said, we always say in the pharma injectable manufacturing facility, what is the most common source of contamination in a clean room? And that's a human being. Because the rest, everything can be automized, everything can be controlled remotely, but you cannot control a human being, because human beings will have microorganisms on their body. They are the common microorganisms, will always be there. Whatever you do, you gown, you perform the best gowning and so on, but still you will move inside those areas and you will take microorganisms inside the areas. So how can we get rid of that? So because how can we reduce the people interface and thereby avoiding the contaminations happening inside the area? So this is the thinking process behind it. So new technologies like isolators and wraps. So wraps is rapid access barrier system, which are fundamentally changing the manufacturing environment of the injectable products. So you have very less people interface. Of course, you still need people for certain activities to be performed. However, they're not in a close contact or in a direct contact of the product. So the chances of product getting contaminated are very, very less. So this technology, if you see in the pictures, here is on the top hand side, you see a conventional injectable line where there are glass doors, and many times operators have to open those doors and do manipulations. If a vial is falling down on the running belt, they have to intervene, go inside with the hands and keep it back or take it out and so on. By doing so, they were also bringing in a lot of contamination. Things have changed with the technology. We have now isolators and wraps where people are still needed to some extent, not to the same extent like we use on the conventional line, but still they do all these operations through Glowport, and the environment inside these cabinets is sanitized completely. or it is decontaminated. That's how we call it. We cannot call it sterilized, but decontaminated to an acceptable level using vapor hydrogen peroxide and so on, so that you get really an environment which is free of microorganisms, and you can do a lot of manipulation without reaching over the product. So that's how the technology is making life easier and also safer for the patients, that you do not end up in a contaminated product going to the market. So that was about the technological part from the manufacturing side. Now let's go to the testing part. So it's not only the technology on the manufacturing side is changing, but also the technology on the testing side is also evolving and changing over the period of time. So until recently, microbial testing was, it was a kind of most conventional type of testing, time consuming. We know that microorganisms take their own time to grow. We cannot force them to grow. Even if you provide them a lot of food, they take their own time to grow. With the evolution of technology, the testing paradigm is also changing. The way how we look at the testing is also changing over the period of time. We are looking now for more reliable methods, but at the same time, faster methods. And this is also evolving. Every year I see there is something new coming in the market, which is making the test more faster, more reliable. Because typically it's always, when I was working in 90s in a microbiology lab, some of our bosses used to say, how confident you are about this result, because these are microbiology, you cannot validate to the fullest extent. So these questions were, always bothering us in those days, because our range of validation was quite large. But now with the new technologies, we can also be very precise, like the chemical analysis we perform on HPLC or GC technologies, and we are more reliable and also more faster. Microbiologists have to apply science behind the test, but use the technology for better and faster assessment of the results. That's the paradigm shift. So of course, the knowledge remains same, the interpretation remains same, but we use technology to be faster and to be more reliable. That's the paradigm change we're talking about. Now let's take a few examples of the specific testing. So I mentioned in the role of microbiologist certain tests, and I will give a few examples how these tests have evolved over the year when it comes to the technology. Let's take the example of sterility. What do we do in the sterility test? We take some X number of vials or ampoules, and we break them open, we filter it, and then test whether there are microorganisms present or not. So that's how we confirm that the product is sterile, so it's free from microorganisms. In 90s, if you see the topmost right picture is the sterility testing device, which I used in 90s, which is a conventional type of sterility test device where we used to, you have a membrane filtration under a laminar airflow with some defined background of zone B and so on. We cut open the ampule vial, put the solution inside, filter it with the vacuum, and then manually cut the filter into two parts, put into two different media, and so on. So this was a conventional way of doing the sterility test. By the time we reach year 2000, we had this, now I ask you to look at the picture in the middle, which is sterility test which uses the closed type, which is called SteriTest device, where there was a kind of automation where the manipulation is reduced substantially. So we do not have to really open the ampule or vial and transfer it manually, thereby increasing the chances of contamination, giving false positive results, and so on. All those things which were in the past happening quite often were reduced substantially when we move from open type of sterility testing to the closed type of sterility testing, which we call SteriTest, which is of course a brand name SteriTest, where the interventions are absolutely less because you directly inject the syringe into the vial by puncturing the stopper, and the machine does everything in a sterile condition. There are media can also be added without making any contamination prone manipulation. So this is how ease of doing it, more compliant way, because we are not bringing in additional contamination while doing the testing, and at the same time it is more reliable and faster. When I think about 2020, here we are talking about rapid sterility test. Sterility test typically takes 14 days of incubation, and you have to wait until 14 days to get a batch out of the testing, and then only you can release. But now we have a technology which is called rapid test using the scan RDI. So this is a technology where you can, if the method is validated, you can release the product within 48 hours. So that's the change. That's the scale of the growing technology in the testing field of the sterility test. This is just one example. If I go to environmental monitoring, the same thing here as well. Environmental monitoring is very important, as I already mentioned before. Traditional way is to use settle plates and air sampling techniques. You keep open the plate in the environment, or use the air sampler, and then incubate those plates over a period of time, 5 days, 7 days, and then you look for the plates, see if there are microorganisms, identify them, and so on. So this is the conventional way of doing this. Now this is also getting changed with the technologies like a rapid test by using continuous biofluorescent particle counters, where the particle counters take this air inside. With the air goes the non-viable particles and also the viable particles. The system is capable of detecting those microorganisms and giving you the absolute number in a more quicker and more reliable way. So this is the future when it comes to the environmental monitoring. Similarly, for the water testing, as I said before, water is an important ingredient or the excipient used in the pharma industry, and it has to meet certain quality parameters, particularly from the microbiology point of view. And detection, and that's the reason why the detection of the contaminants is extremely important in the microbiology department of the pharma industry. Traditionally, we used to have isolation and enumeration technologies. I myself have done a lot of such identification using different biochemical tests, using, of course, in those days also, there were some kits available and so on, but still we were more relying on the traditional technologies involving the identification and enumeration. However, now we have rapid test, which use polymerase chain reaction, so PCR test, ATP bioluminescence test, and so on, which give you quicker, faster, reliable results of these tests. And this helps manufacturer to start their manufacturing at early stage, also to take the results and then release the batches to the market much faster than it used to be in the past. Maybe the last slide on the testing point is about the identification. We do a lot of identification. That's one of the thing I still remember, which used to do also from the university days. Identification of microorganism, performing different tests, triple sugar ion tests, oxidase, catalase, and so on, different sugar utilization, and so on. You have to prepare different media, inoculate the culture, and then wait for the results, and then jot down those results, go to the reference books, find out which test is positive, which test is negative, write it down on a piece of paper, and then come to conclusion which species or which genus or which species these microorganisms belongs to. But this was quite time consuming, although accurate and of course science-based, but quite time consuming way of identifying microorganism. When it comes to pharma industry, we cannot afford such a long time when we have to really identify the source of contamination, because we have to not only identify but to react to the situation. If I'm getting contamination on a line or for a particular product, I need to understand quickly from where this contamination is coming. If I understand that, I can take corrective action. I can stop that source of contamination. And for that reason, identification, or rather faster identification, is very crucial. And this is how the technology is. This is where the technology is playing a role, where we use nowadays, this, maybe some of the universities also using this. I do not know. I have nowadays very little interactions with the universities, but this YTech or Microsec, Multitop, these are some of the automated systems which are being used also to identify up to the genetic level different microorganisms. You know, they use different technologies like mass spectroscopy and so on, but the results are more faster. Of course, the theory still remains same. They use the same theory behind this. The knowledge doesn't go away. Knowledge remains same. The technology is evolving. Technology is helping you to come to a conclusion in a much faster way. So that's how the testing is also changing substantially. So this is more or less about how the technology is evolving conceptually in the manufacturing as well as the testing pyramid. And Nikita, maybe if you allow me, I would like to go to something about the career guidance. Is that okay? Or you have any other?
Yes, sir. Sure.
Yeah.
Yes, sir.
Okay, thanks. So I was told that there are also some students participating, and I thought, why not spend a few minutes to tell them about, you know, what should they do if they really want to get into pharma industry to make their career as a microbiologist. So what are the opportunities, what they have to do? I explained already in the beginning, you know, what is expected from a microbiologist, but to get into that field, there are certain prerequisites which you have to consider if you seriously want to be in the pharma industry. So let's look at those things. First of all, I would say you should ask a few questions to yourself, you know, because I see this, even I myself was a bit confused at the beginning of my career, and that's the reason why I spent a year or so in biotechnology R&D, and then I moved to pharma QC microbiology labs, and then made my career in quality assurance subsequently. But this type of confusions are always there in our mind because we are not clear what is expected from a certain part of the industry or certain part of the research organization, and we end up in entering into a field which probably we may not like. That's the reason why we should ask ourselves, why I have chosen microbiology as my major subject. Is it my interest in the subject, or what else is really was the trigger behind selecting microbiology as a major subject? Am I preparing myself for further education? You know, doing purely a bachelor's degree nowadays is of no use, you know, particularly in India where we have fierce competition. Getting into industry, particularly the big companies, you know, multinational companies and so on, it's not possible that with only bachelor's degree you can enter and do something. So am I preparing myself for further education? Am I going to do master's, or am I going to do PhD, for example? If yes, then what is my interest? My interest is in academics. Is it in research, or I want to do industry job? You know, like I was doing in 90s. You know, I started my career in industry job. What is my interest? These questions you should ask. What opportunities do I have in the industry? So, for example, there are pharma industry opportunities, there are biotech, which I always consider as a separate industry because the way they work and the scope and the scale is completely different. So I always consider as separate industries, so biotech and of course the food industry. So these are a couple of possibilities where from industry point of view, you can think about. And then, am I interested in research or a non-research job? I think this is the most important question you should ask. If you're interested in research, then of course there are possibilities both in the industry as well as non-industry research, but you should be clear about it. You know, if I want to research, then there are different ways to go also in industry and do research. But if you're not interested in research, then it will be more better decision to go for a quality control, quality assurance type of job in the industry. Then coming to the pharma sector, and now I exclude biotech, as I said before. When we talk about the research, as I said, in pharma sector also we have research possibilities. In the basic research, in R&D support labs, also there are independent institutes or company, we call it contract research organizations, CROs, and of course big pharma companies. So all these areas, they need microbiologists also for the basic research. So this is one of the opportunity for those who are interested in doing research, but to be in the industry, so pharma industry. So this is one part of the scope. The other part is the quality control, where you enter as a microbiologist in a manufacturing unit. This is typically in the manufacturing unit or maybe in the testing laboratories. Nowadays, there are quite many private or public testing laboratories involved in the testing of the pharmaceutical products, but more or less related to manufacturing of the pharmaceutical products, where they need a microbiologist, particularly in injectable or vaccine manufacturing areas. There are more openings for microbiologists. If I have to consider the scope, increasing scope, then I would start from API, so active pharmaceutical industry, active pharmaceutical ingredients industry, then comes the oral solids manufacturing companies, then comes the injectable and vaccine medical devices. So the last part, which is injectable vaccines, medical devices manufacturing part, has the maximum opportunities and maximum importance as well for a microbiologist. So think about it, what I want to do. If you're keen on doing your career, maybe you can choose one of these opportunities in the pharma sector. What are the growth opportunities? We always think, I enter as a microbiologist, how long I continue to do analytics in the lab, and it's a fair question. But I can say from my own experience that microbiologists have fair opportunities to grow in their career in the pharma company. The typical end point for a microbiologist to grow in the QC environment to become a microbiology lab head. That's a typical end point where in the big companies, where there are, I was mentioning before, in big companies, particularly in the injectable manufacturing companies, you always have a separate microbiology laboratory with people ranging from 10 to 50, 60 people, and you become head of that function. That's one of the high career point from the lab perspective. But that's not the end point, because typically what I have seen in my career, that microbiologists are best suited for quality assurance function. And quality assurance function has no boundaries. You can grow to whatever extent. Whoever, I've seen quite a many of quality assurance experts in the industry, they have microbiology background. And I always think, why is this so? I think, in my opinion, this is because microbiologists have more of a logical thinking. To be successful in the quality assurance role, you need two definitive characteristics. One is the analytical thinking, and other is the logical thinking. What kind of mix do you carry depends on how successful you are. But I think, in my opinion, it's more about the logical thinking. You have certain data, you of course need to analyze that data, but you have to go beyond that and think logically and then come to a logical conclusion. And I think microbiologists are better placed when it comes to logical thinking. And that's the reason why you have a lot of opportunities in quality assurance functions to grow. I don't know, there is no limit to grow in the quality assurance function. What should I know? Master's degree in microbiology, that's the minimum expectation, as I said before. If I'm really serious in making my career, then in my free time, in your free time, you have summer vacation, and I'm now talking about students, not for the professionals who are joining this, who have joined this call, but for students to use their vacation time, find a temporary training job, which will give you some lab experience. This is very important, because when I hire a person for a lab, I always look for a person who has some hands-on experience for the test which we are performing, because I definitely do not want to spend a lot of time on training of a new or the fresher. Of course, we still do that because we are not always getting experienced people, but it is always an added point that you get some knowledge of the lab experience by doing certain activities. Go to a small scale company, ask them, I would like to work free, I would like to learn, and so on. There are also apprenticeships available in many companies where you will learn how to do the analysis and so on. This is always an added point for you before you apply for any job. Another important thing which people quite many times make a mistake, that they do not want to join a small company. It's a small company, I will not get much future opportunities in this company, and so on. But I think we should not wait for big opportunities. We should grab the one you get first. You can always switch after gaining some experience, because as I said in the previous point, that gaining an experience is very important to grow in the field of pharma QC laboratory. So wherever you get an opportunity, be it a small company, join there, get some experience, and then look for a bigger or the new role. That's very important. Another important aspect is to keep yourself updated about the pharma sector and company. That's the reason why I today in this call I was explaining you how the things are changing. So we are learning certain things in the university, which is of course very important. It's crucial. It's a theory. Without theory, you cannot apply it into the reality. However, you have to also make yourself aware of what's happening around. Read about it on the internet. Nowadays, you have internet available freely. You can read about what's happening in different fields of microbiology, where the research is happening, where the technology is growing, and so on. Talk to your friends, your seniors who have already been working in industry for a certain period of time. They can guide you. They can also guide you what you should read, which type of test and knowledge you should gain, and so on. So this will prepare you when you go for the interview, or even for the person who is hiring you. He will feel more comfortable if you have at least some background of the test or the technologies being used in those pharma companies. Then you might have learned a variety of topics, in BSc, while doing BSc and MSc, we learn a lot, Now I even do not remember which all subjects I learned in 90s as a part of my curriculum in bachelor's or master's degree. We have to keep one thing in mind, that only a part of it is required in the pharma industry, And we have to also understand there are specific needs, and we have to prepare for the interview considering those specific needs. I may be very good in genetics, or I'm very good in enzymology in my curricula, in my university degree, but that's not something the QC laboratory is looking for. What QC laboratory head is looking for is, do I know how to enumerate and to identify the microorganisms? Do I know what are the environmental conditions to grow certain, microbial flora of a human being? These are the questions. So find out what is relevant for the job, and not the theory part which we learn. We learn a lot. And of course, I'm telling this again and again. Theory is extremely important. But when you prepare for the interview, look what the hiring person is looking for and prepare accordingly for the interviews. Another important aspect is to keep in mind that industrial jobs are not easy. It's really, you need to be determined. There's a lot of hard work. Of course, hard work is required everywhere you go. And of course, a lot of patience. You have to have a patience to grow in the organization. You cannot jump from one day to another and become a lab head. So you have to have a lot of patience, determination to be successful in these roles. You have to also keep in mind that there is a world outside your university and college, which is changing much faster than the university. And I'm no way trying to point out that universities or the education field is not changing. In universities and education field is also changing tremendously. And I see this when I participate in such lectures, lecture series or the seminars. I see even the education field is also ever changing. But as I said before, the needs of education field and the needs of industry are different. The pace of the change is also different. And that's the reason we have to also adapt. What are the evolutions happening? And we have to understand, we have to adapt, we have to read, we have to discuss. And if you do not understand, you need to ask. I think that's the only way how we can prepare ourselves for the future or for the career in the pharma sector, if you want to do or if you want to go in that way. Okay, so that's more or less everything. I think I'm on time. It's 13:59 in my clock. So last 1 minute to wrap up.
Sure, sir.
Sorry, say again.
Sure, sir.
Okay, so I was talking about the summary part. Basically, as I said before, it's quite a big sector, not only in the world, but specifically in India. And why I explained this, we are the biggest manufacturer of the generics products. And this situation will even go beyond those 40–50% I mentioned about, and the world is more and more relying on the manufacturing sector of India, particularly pharma. As I said before, it's constantly evolving. That was the topic of our today's discussion. It's evolving continuously. Information technology, artificial intelligence, disrupting technologies. These are going to have very, very big impact on pharma industry in general, and that's why, as a microbiologist, we have to really catch up the speed of those changes. Technology-driven changes need to be adapted. I think this part I have not stretched much about. We have to be really adaptive to the technology. We always get used to certain things. We are used to do certain things in a certain way, and changing from this way to that way is very difficult. And I've seen this even in my career, in my pharma career, when I want my people to change from the, for example, open type of sterility to the closed type of sterility. No, this is very difficult. It takes time. I have to do this, I have to do that. This adoption is very, when the adoption is very low, you lag yourself behind, and then technology goes forward, the needs of the time changes, and then you are leaving yourself behind the technology. So that's the reason why you have to also adapt very quickly. Again, I want to stress, what we learn in the university is 100% essential. Do not misunderstand that. But using that knowledge appropriately in your work environment is crucial. So use the knowledge, but use the technology as well. That's very important. If you start now, you can definitely and easily achieve those skills. This statement is again for the students who are participating in this lecture series. So you are still students, you can still catch up. There is a long way to go. You can definitely do that. And maybe the final statement is that microbiologists are and they will remain an important pillar of the pharma sector. As long as pharma sector is there, the microbiologists' demand and their importance is going to remain there. And with that, I stop here, and I don't know if there are any questions for me.
Thank you so much, Vaibhav sir. So it was a very impactful presentation, I would say, and a very powerful presentation where we went through some technological interventions and breakthroughs, and it generally a connecting link was like highlighted between the microbiology and the pharma sector basically. So the house is open for the discussion. If you have any queries, you can just post in the chat box, or you can just unmute yourself and ask. Anyone with the queries, please?
Yes, good afternoon, ma'am.
Good afternoon, sir.
Yes, sir. It's a pleasure. That's a very nice presentation, and we really gain a lot from it, and it's also a pleasure to understand some new things. Actually, sir, in your distribution of activities, talking of the one that relates to testing, you under the microbiological tests, you put environmental monitoring under microbiological tests, as well as under environmental or under laboratory activities. So I feel that it can go more of laboratory activities than microbiological tests. That's number one. Then another thing too that is very much related to my own scope of research. For example, we are making some new discoveries, through the use of some antimicrobial agents from plant sources. So what is the best method or approach to package them for pharmaceutical purpose? Especially those that has some kind of very high antimicrobial potential against some infectious agents. So what is the major approach that you advise in packaging them for pharmaceutical purposes?
Yeah.
Thank you, sir.
Yeah, thanks for this question. I think it depends on what type of the final product you're looking for, because there are set rules, there are set guidance available in the pharma industry. So for example, if you want to make a drug product out of those substances which are meant for oral solid dosage form, then you need different environment. But if you want to use those substances for injectable manufacturing, then you need more stricter environmental conditions. So it really depends on what is the applicability of the final product. So it will depend on that basically. Did I answer your question?
Yes, sir. No, I'm just looking at the realm that, for example, now in my research lab, we are working on the use of some plant extracts synergized with chitosan, and this has some antimicrobial activity against some enteric organisms and few other organisms. So under that context, we are doing some kind of drug development, and under that context, is it advisable to package it as capsule or as tablets or in powder form?
So your question was more from the formulation point of view. Okay, now I understand.
Yes, thank you. Yeah.
I think this will again depend on what type of pharmacological activities you're looking for. And unfortunately, I'm not an expert in the formulation development, but formulation development people can guide you depending on the applicability of the drug substance. They can tell you whether this can go as a capsule or this need to be an intravenous injection and so on. So those are the people who can guide depending on what pharmacological activities you're looking for out of that drug substance. But typically, for antimicrobial substances or anti-infective substances, you can have all type of different formulations. So for example, you get let's take a simple example of amoxicillin. So you get amoxicillin also as a tablet. You also get it as a suspension where you make liquid of it and you take the liquid as a suspension. You also have injectable product. So the applicability of the drug product depends on the condition of the patient and also on the final impact of the drug substance on the patient.
Okay, thank you. Then on the aspect of environmental monitoring, I just want a concise response to that, sir.
So
environmental monitoring. Under environmental monitoring, you classify it under microbiological tests as well as microbiological activities. So I just want a brief.
So environmental monitoring, as I have marked it under the microbiological test, but it is because the part of it happens in the microbiological lab. So that's the reason I highlighted it under the test. But basically, it's about monitoring the manufacturing environment. So basically, what we do is we expose the agar plates into the manufacturing environment. We do the air sampling. We do the personal monitoring, we call it. So we take swabs from the from the people who are working in the environment, and then those plates are then taken back to the laboratory. They are incubated at certain temperatures for a certain number of days, and then we count the number of microorganisms growing on that. So it's a combination. It's also the activity performed on the shop floor, but at the same time, later on, you have to incubate and read, and that's why it's part of the laboratory test as well.
Okay, thank you, sir.
I have one question. It is little different from today's lecture. Can you throw some light on what is the status of plant-based bioactive molecules in the pharma industry?
Plant-based substances are also evolving, but as I said, the technology is more and more going towards, how can we re-engineer the cells itself, So we are talking about the CAR-T therapy, radioligand therapy, gene modification and so on. So rather than treating the patient using the external drug product, the thinking process is going into a direction that we make the changes in the body itself so that body will start reacting to those situations. So that's how I see, of course, the importance of plant-based extract substances will not go away, because these new technologies are really more focused on difficult indications. So for example, more and more on cancers or the diabetic conditions and so on. But for the normal, normal type of diseases, day to day, when we face those diseases or the conditions, the importance of plant-based extract or the substances coming out of the plant-based extract will still be there.
Okay, thank you.
Thank you, sir. So any more queries from the audience?
Hello. Yeah.
Yes, ma'am.
Yeah. This is Kirti Apare. Actually, I just have a question regarding the as Mr. Vaibhav sir said that the stainless steel sterile preparations, how because R&D scale it is available, but for commercialization of any product, the sterile bioreactor of what capacity has been used in the industry?
Quite huge, I would say, quite huge, because I know what you're referring to. In the past, these small disposable ones were being used in the biopharma industry, where the scale was quite low, or the small scale and so on, or maybe mostly in the R&D function and so on.
Yes.
But this has changed since the COVID time. This has changed, and now we really get huge bioreactors. So 20 KL, 50 KL, 100 KL capacity bioreactors are available nowadays.
Okay. Thank you.
Thank you, sir. Any more queries, please? So, sir, I have one quick question. Basically, I came through a dilemma process of students, like pharma students, they want to adopt only pharma sector, like as graduation or post-graduation. They are not well aware about the microbial pharma connect. So basically, according to you, how would you suggest a type of micro pharma connect, and how successful it would be when a student think of connecting these type of synergistic combinations, basically?
So if I understood your question correctly, when you say pharma student, means those who are studying pharmacy, right?
Yes, sir.
But I think in the course of pharmacy, they always have microbiology as one of their subject, right?
Yes, sir.
But this is always the case, that you try to focus more on your core subject, and their core subject is always, formulation, pharmacology. Sometimes, those are the typical subjects they would like. Pharmaceutical sciences, mostly the formulation part of it. So that's the reason why for them microbiology is, one of the subjects, not the most important subject. But of course, they do understand, particularly when they enter into pharmaceutical companies for their job, they do understand, particularly when they're working in the sterile injectable manufacturing, they realize that, yes, microbiology is an important subject, and probably I should have learned more, or maybe learn more seriously those subjects, because it's going to be very useful for them also when it comes to their day-to-day life. So I think it's up to the teachers and the professors to really give emphasis that microbiology is also important, particularly for the sterile injectable field, which is quite large in the pharma sector as well, where microbiology is as important as the pharmaceutical sciences.
Thank you so much, sir. So we end our session today, and it was a very powerful session, and I too learned a lot of things, and what was the technological shift from various type of sections. So thank you so much, sir, and we'll be connecting in future for your further lectures, and we'll be inviting you for that. And if anybody wants to have some further questioning and all with Vaibhav sir, so I'll be just sharing his email ID with you, and also the presentation. So we'll be sharing a presentation with the audience of MBSI, and all the students. They will be like having the YouTube streamlined sessions and all. So thank you so much. Thank you, everyone, and keep growing and keep glowing. So we'll be meeting in the next lecture on Sunday Talkies. Thank you so much. Thank you.
Thanks a lot. Bye.